welcome back to my course Aspects of Biochemical Engineering. Now, last couple of lectures I try to concentrate on kinetics of homogeneous reaction. Now, today I want to discuss one very important topic, that is different type of reactors. Now, a question comes, what do you mean by reactors? Reactors means a kind of vessel in which the reaction takes place. Now, that means it is very simple, that a container, a vessel means container, when we put different reactant where the reaction takes place, that is called reactor. Now, what do you mean by bioreactor? Because we have another term, because since we are this particular course is on biochemical engineering. So, bioreactor also it is same vessel in which the reaction takes place in presence of some either from biomolecules. So, maybe it might be living cell, it may be non-living cells. I can give the example. Suppose some reaction take place in presence of living organisms, we call it a biochemical reaction, or it is suppose the reaction carried out with the help of some enzyme which does not have life, but enzymes that is also we consider as a bioreactor. So, if you look at here, the reactor is the heart of chemical processes, because because here where the reaction takes place, am I right? And chemical reactor is a vessel in a chemical process or plant where the chemical transformation or reaction takes place to generate the desired product. So, it is very simple. Say, in the vessel in which the reaction takes place, that is called chemical reactor. This is like this. Now, if you look at the bioreactors, deals with organisms or biochemically active substances derived from such organism. If the reaction takes place with the help of this organism or biochemically active substances derived from the organism, because as per example enzymes can be are secreted by the living cells, maybe plant cells, maybe microbial cells, maybe you have animal cells. So, if the reaction takes place with the help of enzymes, that also we can consider as the bioreactors. Now, question comes, what are the bioreactors and the unit operations? So if you look at this is the bioreactors, what do you call the heart of the chemical process. After the reaction is over, we passes through the separator. So, any biochemical or chemical industry, we have different separator process. What do you mean by separator process? Separator process means where the solid is liquid separated from the liquid, or liquid separation also will be there. Then there is the involvement of the heat exchanger, because as you know, any kind of chemical reaction or biochemical reaction, that depends on temperature. So, we shall have to maintain the temperature. So, we require the heat exchanger. The utility, different utility that also present in different industry. Utility like we might be requiring the soft water, we might be requiring some steam requirement. All this comes under this utility. Then process control, that is another important to operate the process, that we shall have to maintain the temperature, we shall have to maintain the agitator speed, we shall maintain that pH of the solution. Some cases we shall have to maintain. So, these all are under this unit operations. And as for example, for controlling the temperature, we shall have both the cooling and heating arrangement. For maintaining the pH, we shall have both the acid and alkali tanker. Your pump should be connected with that. As the pH going down, you have to add some alkali to it. As the pH going out, you shall have to add some kind of acid to control the pH. So, these are the things that we have. Now, let us see what is the classification of the reactors. Now, we look at the classification of the reactors, this may be classified in 3 different ways. One is called mode of operation, the geometrical configuration, the contact pattern between the phases. So, mode of operation. Mode of operation means the three different mode of operation we have. One is called batch process, another called fed batch process, another is continuous process. Now, question comes: what do you mean by that? Batch process. Batch process means if you look the batch process is like this. Suppose this is the reactor, and here you have a starter, and this is liquid. So, we put the substrate here. We want to react A to B. So, we put the substrate A, and then at a time we shall have to put the substrate at a time in a batch reactor. This is the batch reactor. At a time we shall have to put that substrate, let the reaction take place. After the reaction is over, you take the material out. In between, you are not putting any material inside the system. Now, if you look at the fed batch, what you call fed batch is similar to that reactor, whatever I have shown you. That only the thing we start with small volume, and then slowly we increase the volume. Fed batch, different batches we put it like this. Now, what is the basis of that? Why we do that? Because and how we do that? If you look at like this, this is the substrate concentration Ca. So, if you consider this is V1, am I right? This is V1, this is V2, this is V3, this is V4, this is V5. So, you know that when it comes here, then you stop the operation, and after the reaction is over, you take the material out. So, what is the fed batch? You give the feeding, but you are not taking anything out from the reactor. That is why we call it fed batch reactor. Now, here I can give the example. Suppose your substrate concentration keep on decreasing with respect to time. This is with respect to time. Then again you are giving a feed here, so that you increase the. Now, this level of substrate should be much below the inhibition level. What is inhibition level? Inhibition means every substrate above this concentration which retard the chemical reactions. If retard the chemical reaction, that is undesirable, then your product formation will be less. So, we want to operate the system below the that inhibition level. So, what we do initially, that we fix that concentration much below the inhibition level, let it go down. Then after certain time, again we give the feed to rise at the same level, again it goes down like this, again rise it, again goes down like this. This is how V1, this is V1, V2, V3, like the different volumes we get. Now, we can have another pattern here, that we can suppose Ca, this is repeated feedback, and this is another we can do. This is decreases like this, then increases. Then we slowly food in such a way, the rate at which the substrate consumption take place, at the same rate we are feeding it, and we feed up to this level continuously, and then we stop the feeding, then it goes down like this. So, there are different way we can operate the fed batch. Now, why you go for the fed batch system? The fed batch system is applicable where there is a substrate inhibition. In case of substrate inhibition, we prefer the fed batch reactor. We will discuss when we discuss some different processes. I will discuss in details. Now, another reactor, we have the continuous reactor. What do you mean by continuous reactor? Continuous reactor. Continuous reactor means, suppose this is the reactor that we have here. We have a starter. So, there is a continuous inflow and continuous outflow. Now, here I want to point out one typical thing, that whenever we operate any kind of continuous process, first usually we operate in the batch mode. Suppose we want to carry out the A to B. We want to operate in the batch mode. Let the reaction take place. When rate of reaction is maximum, then we start feeding this continuously. We take some product out continuously. This is how the continuous system has been operated. Now, continuous system may be of two types. One is called CSTR, another we call plug flow reactor. PFR. plug flow reactor. Okay. So, two type of the plug flow reactor looks like this. We will discuss in details. The difference is that in case of CSTR, this is CSTR, it is a continuous start tank reactor. A continuous start tank reactor means you have a starter and continuous inflow and continuous outflow, and this is very easy to operate. Now, if you look at this is the plug flow reactor. In plug flow reactor, you are giving the feed to one end, and it is something similar to the tubular reactor. So, so one end you are feeding and then you are taking. So, when liquid flows like this, there should not be any kind of back mixing or axial mixing take place. There might be radial mixing is okay, but that back mixing does not take place. But here we have back mixing, but here we do not have any back mixing. So, so we have here the the different mode of operation, the batch, fed batch and continuous. Then if you look at the geometric configuration, we have tubular and agitated tank reactor. Tubular means it is kind of tube, liquid is flowing through the tube. Agitated reactor, I have already shown there is a agitation that we have. And then contacting pattern between the phases, we have the pack bed reactor, we have fluidized bed reactor, we have trickling bed reactor, we have bubble column reactor, we have air lift reactor. So, different type of reactor. So, this is the heterogeneous reaction. Heterogeneous reaction means more than one phase is present in the reaction mixture. So, this I shall discuss in details when I shall discuss the individual processes. Let us look into this that batch reactor that we have. Batch reactor is the reactants are charged into the vessel at the beginning of the operation, and the products are discharged at the end of the operation, and during this process there is no feed addition or product withdrawn. So, this is exactly what I told, and batch system is very easy to operate. Just you take the substrate and put it in the reactor and let the reaction take place. After the reaction is over, you take it out. This is called batch process. Now, the chemical reaction, this is application is the fermentation of the beverage production, chemical industry for dyes and ink production and wastewater treatment process. It is largely used. Now, let me talk about this fed batch reactor. Fed batch reactor is a tank in which the reactant are charged initially, and limiting reactant is added continuously during the operation, and no product removal during the process. So, I told you that in this reactor we start with small volume, then let the reaction, then slowly we feed this liquid like this, and until this is the final volume. When its final volume is coming, then let the reaction take place. After the reaction is taking place, you take the material out. So, but during feeding you are not taking out any product from the reactor. The application is the chlorination of a liquid, pharmaceutical industry. Particularly, I can mention the Baker's fermentation process. Baker's fermentation process is largely used. Now, if you look at the Baker's, the 1 mole of glucose produce about approximately 0.5 grams of substrate cell mass. So, naturally, if you use more your system, you utilize more glucose or more sugar, you will get more cell mass production. So, naturally we want that how this reactor can use more substrate, because we know if after certain substrate concentration it has some inhibition effect. So, but to consider all these things, we find that fed batch is a process through which we can increase our Baker's production to a great extent. Now, let me talk about the continuous process. Continuous process is the vessel in which the reactants are fed continuously and products are withdrawn continuously. Only the thing I highlighted before, That continuous system, when you operate, first we operate in the batch mode, and after the when the rate of reaction is maximum, then we continuously put the reactor in and continuously take the product out. The reactant take place over space, and that is the application is the chemical industry like Harvard process, biological industry largely used, and brewing, antibiotics and wastewater treatment process, we use this particular process. Now, pictorially that this process looks like that batch process, we put the material at a time. After feeding, we are not taking the material out. After the reaction is over, we can take the material out. But in the fed batch process, we small feeding we should have to give with respect to time. But at the here also we cannot so there we have a feeding continuous. Here we don't have a continuous feeding, but here you have a feeding here. So until unless the final volume is reached, we do the feeding. Then we stop the feeding, let the reaction take place completely. Then we take the product out. In the continuous system, the continuous inflow and continuous outflow from the system that take place. This is how we can explain it pictorially. Let me talk about this tubular reactor. Tubular reactor is kind of plug flow reactor. This is a plug flow reactor. I told you it is like this. This is the tube, and you pass this, and it will flow like this. Now, basically, the tubular reactor is plug flow reactor is that it is kind of plug flow means is call it the plug flow. That is nothing but we call it piston flow. What is piston flow? Because we know if you have a piston, when it when you have a syringe, we have a that we have when you put this shaft forward, then the liquid is moving. When it's moving, there is the velocity across this liquid, across this cross section will be more or less uniform. But when liquid flows through this tubular reactor, we have gradient of this velocity, because we find the liquid close to the cell wall with the wall of the tube is the less velocity as compared to that of the center, because there will be friction between the wall and the liquid. That is why the velocity reduces to a great extent. Now, reactor consists of hollow pipe or tube through which the reactants flow, where the reactant moves as a plug flow along. Now, piston flow basically we consider as the ideal flow. Ideal flow is kind of very rare to happen. So, you know that we can, what if you make the flow the turbulent flow, then it tends to piston flow, not exactly piston flow, tends to flow. Then velocity gradient across the reactor will be minimum, because that velocity gradient across the reactor will be minimum if we flow pattern, if we change from laminar flow to the turbulent flow. Now, concentration of reactant that varies along the pipeline. So, if you plot the concentration, you will find keep on changing with respect to distance. No axial mixing, but only the radial mixing. What do you mean by radial mixing? I told you this is the tube. If your tube is there, so the liquid can have the radial mixing, that in a circular direction, not in the axial direction. This axial direction is not permissible, but radial mixing is permissible. The homogeneous, if you look at the homogeneous and heterogeneous reaction, it is used application, and continuous production and high temperature reaction, this is used. Now, this is how pictorially this process looks. This is tubular reactor, this is tube. You can see how well you put the feeding one end, you take the product in the other end. Now, CSTR is a kind of reactant, and products are continuously added and withdrawn while the content within the vessels are vigorously stirred with the internal agitation. The cascade CSTR can be used for the higher conversion of reactant. What is cascade reactor? Cascade reactor means if you have the CSTR in series, because this is CSTR. If we connect with a series, then we call it cascade reactor. This is we call it cascade reactor. Now, application is biological industry such as the brewing, antibiotics and waste treatment processes were largely used. It is a continuous charge tank reactor looks like this. This is the vessel in which the reaction table. There is a starter, and the purpose of starter to maintain the homogeneity in the reaction mixture. We put the feed in one end and take the product in other end. Now, let me discuss the packed bed reactor. Let me show you that packed bed reactor. Suppose this is a packed bed reactor, and here this is the tubular reactor, the cylindrical reactor, and this is perforated disc, and here you pack the solid material like this. They are lying one after another like this. What I call packed bed reactor. So, you pass your substrate and you take out the product like this from this. This is the packed bed. Now, the interesting feature is that in the packed bed reactor, solids they will touching with inner the packing. This is the packing material. Am I right? This is called packing material. Packing material. Now, they pack to one after another due to the gravitational force of attraction. Now, when you pass your liquid due to the packing, what will happen? This portion where the solid they are lying after another, liquid cannot penetrate this, because the liquid will flow wherever there is a void space. Then in the void space liquid will go. Liquid cannot go where the solid they are touching with each other. So, what I want to say that suppose this is the solid matrix, and whole portion of the solid matrix will not come in contact with the substrate. Suppose we want to do the immobilization of the enzymes, and enzymes are immobilized on the particular solid surface, and whole surface the enzymes are. Now, if the other the solid material they are touching with each other, this portion whatever enzyme is there, that will not come in contact with the substrate. So, that is the problem with the packing bed reactor. And not only that, in case of cell mass growth, when you do the immobilization of the cell will multiply, and when cell multiply, what will happen? This layer will be becoming thickening, and when the layer is becoming thickened, then what will happen? The pore size of the particular pack column will be reduced. As the pore size reduces, then water flow will be affected and liquid flow will be affected, and then there will be the channeling of the liquid flow. If there is a channeling of the liquid flow, then rate of reaction will drastically reduce. Then you have to stop the process and regenerate the system again. Now, let me tell another reactor, what we call expanded bed reactor. Now, these solid particles, they are lying with each other. Am I right? They are lying like this due to gravitational force of attraction. So, this is like this. Now, if you increase the velocity, this is substrate is coming, product is now if you increase the axial velocity, the axial force if it increases, a time will come, this will overcome this gravitational force of attraction, then particle will be separated from each other. What we call expansion of the bed will take place. Now, as soon as the expansion of the bed will take place, then whole portion of the solid matrix will come in contact with the substrate. Then naturally your rate of reaction will increase. So, in case of expanded bed reactor, the expansion of the bed takes place. Now, in case of fluidized bed reactor, this velocity will be much high, and since the velocity is high, this particle separation also very high. There also the expansion of the bed also very high. So this is like this. This is the how we differentiate that. So packed bed reactor, if you look at that, a vessel filled with catalytic pellets larger than 1 centimeter to avoid the excessive pressure drop and the reacting fluid passing through the void space between them. Pumps can be used to make the fluid move through the packed bed. Application is wastewater treatment by using then immobilized cell system, then multi-phase reaction and pharmaceutical industries. Then examples is like this. This is the packing bed. We sometimes we recycle back to have more conversion efficiency of the process, and we take the product out from the top. Now, fluidized bed reactor I have already explained, only I want to highlight here. A vessel filled with the fine particles smaller than 500 millimeter, and that are suspended in the upward flow fluid at the high enough velocity to suspend the solid. And what is the typical velocity is that 6 to 20 meter per hour, and what is recycle ratio is 5 to 500, and expansion of the bed is about 30 to 100%. Now, in case of expanded bed, it is 30 to 40%, and now here it is about 100% expansion take place. The multi-phase system with biocatalyst, food processing industry, chlorination, olefins, alkyl, this is largely used. Now, this is how the fluidized bed occur. You can see the particle, they are separated from each other. Now, trickling bed reactor is something similar to the packed bed reactor. I do not like to discuss again. The bubble column reactor is a very simple reactor. I can give the example that here this is suppose this is the column, and what we do, there is a sparger here. We put a sparger, and this is the liquid, and this bubble will go like this. So you have cell suspension here that will react there. The vessel filled with liquid and gas as reactant and feed from the bottom and move upward in the form of bubbles. Liquid reactants is feed from the top and withdrawn from the bottom. That application is that single cell protein production for which cheese whey it is used. Now, this is largely used in the microalgae algal fermentation process. This is how it looks. This is what we call a sparger, that you can see the sparger, and this is how the bubbles are moving upwards. Now, then air-lift fermenter is we have here the air-lift bioreactor are similar to bubble column reactor, but differ the fact that they contains a drop tube. So, in case of bubble column reactor, we don't have any drop tube. Here you have the drop tube. So, when you do the aeration here, the bubble will go, and some of the bubble will recycle back here like this, and most of the air that will go out of the system. I shall explain pictorially in the next slide. Then application is the photobioreactor for algal cultivation. Largely we find it is very good for the growth of algae, and aerobic bioprocessing technology and single cell protein production. This is different types we have, as I told you here. If you look at that, if you do the aeration at the bottom, and this is the drop tube we have inside the air drop tube, then air will go out, and some of the air, due to the dragging force, it will comes in this area, this annular area, and it will recycle back this. Then another way we can do that in the outer, here we is sparging, and it will go out, and some of the air will come this way, so that more mass transfer will require. And this is how we can done externally. This we can recycle like this, that will take place. 3 different way the mode it can be operated. Now, another thing we have, that is the membrane reactor. This is also very important. This is used for the dehydrogenation of ethane, production of monoclonal antibody and wastewater treatment process. Now, you can see that when suppose the A reacts to give B plus C, so you have different colored shade here. So, due to membrane, you take out suppose you are interested for the product red color. So, red color you diffuse out through the membrane pore, and you can get other than this from the mainstream. You can find out this. So, that construction of the material is very important, and that of the reactor is very important. It should be flexible and durable and non-toxic to reactant and product, resistance to chemical and metabolic products of the organism, resistance to withering, low cost and available, easy of fabrication and corrosion proof, and high transparent. So, high transparent in case of photobiological reaction, because we want to use the light energy for the growth of the microbial cell. Now, for construction of material, I can give the typical example of chromium. If we give the chromium in the stainless steel, then there is acid resistant property of the stainless steel increased to a great extent. Usually, we use the SS304 for our day-to-day requirement. For in the fermentation industry, we recommend SS316, sometimes SS316L, that is we use for pilot pen and large scale operation. Now, this is the lab scale. This you can see that this is the lab scale, and this is the commercial scale operation, how it looks. Now, this is the reactor, how it looks. A reactor will be having and so many things you can see in the reactor, we shall have the agitator, we have the steam for the sterilization purpose. This is the harvesting line that we can, after the fermentation is over, we can take it out. Here the pH probe, temperature probe that can be inserted. Here is the dissolved oxygen probe that is interested. The cooling arrangement we should have, that cooling jacket we have with a nutrient we should add like this. And this is the motor. Through the agitation take place, acid and base is added with the help of pump. Pressure gauge is used to monitor the pressure of the particular reactor. Now, this is the different accessories that we attached with the reactor or the bioreactor that I already pointed out before. I don't like to discuss more. So, in this particular presentation, I try to give you this detailed information of the reactor and what are the different types of reactor is there and what are the different accessories that are present in the reactor. So, in the industry we use 3 different type of reactor. We use the batch reactor, we have fed batch reactor, we have continuous reactor. Then we have seen the packed bed reactor. Packed bed reactor may be of other types. We have expanded bed reactor, we have fluidized bed reactor, and other type of reactors we have, as for example that bubble column reactor, air lift bioreactor, where this is used where the shear force is very less, because in case of normal fermentation process, if we use the mechanical starter, there will be kind of shear force. So, some of the living cell, they are very sensitive to the shear force. So, there we cannot use the mechanical starter, there we can use the bubble column reactor or air lift fermenter. So, this is I hope you will get you get some information on the reactor, which is used both chemical and biochemical industry. Thank you very much.